This is Stem Cell Manipulation by Aidan Matson, a multimedia project. Introduction. In the field of biotech, one of the premier fields is that of stem cells. This is due to their possible medical applications and some other uses in future biotech fields. The reason why they are so important is because of their unique traits. Now, these unique traits and their potential applications have also drawn my interest, and this is why I started on this project. Cellular biology. Now, to understand a stem cell, we must first look at the overall cellular biology of a normal cell. I mean, although stem cells are, they are pretty much this cell here. They are not very unique. To understand the stem cell, we must look at these some of these basic elements that are going to be utilized in their diversification. We must look at their membrane and the nucleus being, at least these two organelles being the foremost in importance. The membrane. The membrane is very important to in terms of cellular manipulation, as its purpose is to keep out foreign objects and keep out unwanted things. So obviously, if we're going to introduce DNA into the cell, we have to go through this membrane. Now, this membrane is made up of phospholipids, and it's made up of phospholipid bilayer, to be more specific. And in this bilayer, there are also proteins and other structures that comprise it. The other main organelle that we are focusing on is the nucleus, and the nucleus is the central element of a cell, and it pretty much, its job is to contain all the DNA and control the cell. Now, we'll need to look at this because when we introduce the DNA into a stem cell, it's going to have to go here to be integrated. The nuclear membrane. The nucleus is surrounded by a double phospholipid bilayer. This is very similar to that of the cellular membrane, although its system of pores is slightly different, with these things called nuclear pores. Now, a nuclear pore is incredibly important because it determines what goes in and out of a cell. And, nucleus. And what one thing has to have to get into this is a specific string of amino acids, and these amino acids allow them to enter through the nuclear pore. And these amino acids are attached to proteins like RNA or DNA polymerase, and then these proteins then enter the cell nucleus, and then do their assigned tasks. Now, in conclusion, to understand cellular manipulation, we must first look at these, and understanding these cellular biology can really help us understand stem cells and how they diversify. Stem cell biology. Now, a stem cell can be thought of as a cell specifically meant to make other cells. Now, this might be a bit confusing, but stem cells don't have any kind of specialization. They are mostly undifferentiated cells. The point of them is to replicate and then differentiate into different kinds of cells. Now, this can be achieved through natural means and through some artificial means. We'll get into that later. Now, as a continuation, while stem cells are fairly undifferentiated, there are different types of stem cells, and this should be noted. This should be noted when discussing this topic. And some stem cells are more or less compatible with other kinds of cells, or at least, well, not as much with pluripotent cells, which are what stem cells are, but as with cell types. Like for example, certain stem cells can't take on certain roles later in life. But getting back on topic, stem cells can be generally divided into two groups. You have your embryonic stem cells, and you have your adult stem cells. And adult stem cells, what I mean by this is a cell that you would find in your body. It's not something that you'd find in an embryo, but something you'd find in your body. Now, embryonic stem cells, they're like the really undifferentiated stuff. They are cells that they can become anything. They can become anything and take on any role. But this does not mean they are quite they're at least infallibly undifferentiated. There are several types of them, and these are the ectoderm, the mesoderm, and the endoderm. And all of these actually have physical locations in the blastocyst, being on the outside, middle, and then inside of the blastocyst, respectively. Now, adult stem cells are slightly different from embryonic stem cells, and this is because they are actually significantly differentiated. This is to the point where certain adult stem cells cannot take on the roles of other adult stem cells. And they come in several flavors. Like, you have your epithelial stem cells. This is the stuff you'll find in your skin. Well, it'll at least replenish your skin and your intestines. You also have your bone marrow, which is your hematopoietic stem cells. And these are what will replace your blood, a lot of that such. And you'll also have your neurological stem cells, which are for your neurons and for your brain, and to replace it and repair that. Now, in conclusion, understanding these different types is key to understanding a method of cellular diversification. And these cells are derived from their predecessors and then subsequently diversify in their own turn. And understanding these different types is necessary to understand how to utilize them properly. Now, cellular diversification. Now, stem cells form the basis of all cells in the body of an organism. And stem cells are pluripotent and have the result to be able to change into many different cells. And this has to be utilized in some way. But this has to be utilized, and so they have to be able to diversify. At least the body has to have a means of changing these cells to do specific tasks. Now, this is through the use of enzymes very commonly, and these create cellular progenitors. Now, these enzymes, a good example of this would be BMP6, which is located in the bone marrow and it uses to help regulate the growth of these stem cells and the rate of their population growth. And so these create the cellular progenitors, which are kind of they're like another step down. They are a kind of diversifiable cell. It's just like another step down. It's a bit complicated, but the point is that these are used to help regulate. Enzymes are used to help regulate stem cell growth and such. Now, the way the stem cells diversify is through asymmetric diversification. Now, this is where the stem cell, instead of dividing and becoming two of the same cell, it divides and becomes it, and one or both have one has a different makeup as the other. Um, this can be activated by any number of things, like minor conditions in the area surrounding it, like for example maybe heat, but it's primarily done through the use of enzymes, and this use of enzymes helps regulate these. Now, just kind of as a transition, I guess, from the natural to the artificial, I think it's worth addressing viral infection, because a virus is actually very similar to an artificial manipulation of a stem cell. And while these aren't necessarily on stem cells, they're very similar in nature. Now, what a virus does is it goes into a cell, injects its DNA, and then has a cell produce more viruses, adapt itself, if you will, to its own purposes. Now, this is very, very analogous to the methods and results of transfection, which we will get into in a little bit. Now, before we address transfection, we must address electroporation. Now, electroporation on its own is not a method of cellular diversification, but it can be seen as more of a method to introduce and change a cell. It's not on its own, but it's like a supporting element. If you were to imagine a courier system or a postal service, this is the postman, and the package is DNA, and the overall method is the postal service. While this is simply just an element of that, this helps deliver that package to the cell. Now, transfection is this overall postal service, if you will. It is the method of infecting would be one word to use, but introducing DNA into a cell and then having the cell change because of that. And this is typically done with electroporation, but there are other methods, as you can see with the diagram. Now, once this DNA is introduced, it is then integrated. Now, the DNA is integrated by transcription factors, and these transcription factors are usually there to help repair and restore DNA. Now, when they see they don't as much see it, but they can chemically detect it. Once they detect one of these DNA strands that's loose and just floating in there, they decide that it's obviously damaged. It needs to be repaired. And so what they do is they attach this to the other DNA in there, to a loose end, and once that's done, the DNA starts going to work when the RNA polymerase comes in and then copies that down into RNA, producing new proteins and such. And this is pretty much how you change the cell. This is the change in the cell occurring. Now, in conclusion, it is key, and it's incredibly important to understand these various methods of cellular manipulation, both natural and artificial. It's just how you do it. And with these, you can understand the implementation and usage of these stem cells in treatment. These methods have allowed many of the developments in the field of medicine. There are many potential applications for stem cells. The primary applications, at least seen through the modern biotech community, appear to be focused around medicine, and this is because they have many potential uses in this field and could possibly change it in incredibly significant ways. Now, one of the primary uses of stem cells that's actually being used today is bone marrow replacement, and pretty much this involves taking the stem cells from a donor, and then after someone undergoes chemotherapy, they're reintroduced or introduced to the bone marrow and help replace lost cells there and help expedite the recovery of this person. They can also be used, at least in theory, to treat heart problems like cardiac lesions. And just by regenerating certain elements of such tissues, they could theoretically change such diseases and possibly even eradicate them. Now, stem cells are not without their controversy, and this primarily surrounds the embryonic stem cell harvesting, at least. This is because harvesting such stem cells typically involves a fetus, and this fetus, well, they do involve a fetus, and this fetus is terminated during this, and because of this, it is quite controversial, just due to the fact that reproductive rights and such are fairly touchy here in the United States. So anyone going into this field or wishing to study this topic in more depth should take this into account, as these simple elements can be very important to your considerations. In overall conclusion, stem cells are an incredibly important part of our cellular biology, and this is due to their ability to transform and become different types of cells. Now, this has also sparked much interest in the biotech community, and as such, they have made significant developments in the fields of medicine and in several of the other fields with these cells. And I predict, personally at least, that these stem cells as a whole, will become a very important part of the future, especially in the biotech community. These are my picture sources. These are my academic sources. Thank you for watching. This has been Aidan Matson presenting Stem Cell Manipulation, a multimedia project.